Please join me in welcoming to the Distinctive Voices podium, Dr. Regina Reagan.
All right, well, thank you so much for joining me this evening. It's my pleasure to present my research. The title of my talk is Intercepting and Decoding Bacterial Communication. If we look at bacterial signals, it can tell us about the presence of an infection. But if we actually look at these chemical signals and try to understand them in more detail, it can also tell us the appropriate drug for the infection. And based on that work, understanding the stress response of the bacteria to an antibiotic, it's provided insight to us to also use bacteria to tell us about the presence of toxins in the environment. So we can detect toxins in the environment well below levels in which cellular damage occurs. So they're kind of an early warning of toxins. So in that sense, we use them as basically a microbial canary in the coal mine. So many of you are likely familiar with how we detect radio waves from space with satellites. Right? So we take these waves and they provide information on what's going on in space. So similarly, if we scale down the electromagnetic spectrum to visible light, we can actually use these waves, how light scatters from molecules, to tell us about the molecules present. But in order to do that, we need to scale down our antennas to very small length scales. So what we use in my group are gold nanoparticle antennas. So as you can see here with the illustration, so we're basically assembling these gold nanoparticles in which the gap between the nanoparticles is about the width of a strand of DNA. So if you've been to the doctor recently, typically, I recently had COVID, a summer case of COVID about a month ago, and they determine your lung health by using a stethoscope and listening to your breathing. But new tools are emerging in understanding the signals from bacteria, and most of it is looking at the genes in the bacteria, right, to tell us the presence of an infection and then the appropriate treatment for the infection. So typically you'll see this kind of analysis in a hospital, like UCI Medical Center. So the problem with that is that usually these tools are not used because they're expensive, they're time-consuming, they require specialized expertise. So what we're working on is to make a simplified lab-on-chip process where you can just use a regular microscope and use machine learning analysis to look at the signals from these bacterial communities in response to an antibiotic treatment, and it can tell us if the antibiotic is going to be susceptible to the antibiotic or resistant. And the idea there is to be able to have these at low enough cost so we can distribute them throughout urgent care centers, right? And they can be fully automated and easy for a regular lab technician to use. So because of the fact that antibiotics are overprescribed, it leads to this antimicrobial resistance. And the literature shows that basically emergency rooms, urgent care, and telehealth centers are the ones that are the higher prescribers of antibiotics. And that's the need for this technology. This is some work done at Technion by the Kashoni group, and it's kind of an interesting experiment that they did. So basically this Petri dish is about 4 feet long, and then they show that, okay, at the ends of the Petri dish they have agar, which bacteria can grow, and they introduce the bacteria at the two ends of the dish. Level 1, that's the typical dose of an antibiotic to kill about 50% of the bacterial cells. So what they show is that they have a video, so you can Google it if you're interested, it's very interesting. So you can see that at the typical dose that will kill the bacteria, you can see that there's a few colonies that have mutated that are now resistant to this antibiotic dose, and then they can grow. And then when you go to 10 times the normal dose of antibiotics, you can see again there's some mutants that are growing, and then they're gaining resistance to the dose 10 times higher. And then this just goes on 100 times higher and up to 1,000 times higher. So they can look back and they can sequence these strains and understand more about the resistance profile. But the issue is, so as we put more antibiotics in our environment, then we have more bacteria that are going to become resistant to antibiotics. And so it only took 11 days for this original group of bacteria to become, to acquire resistance at 1,000-time higher dose. And as you know, if you take in antibiotics, usually they make you a bit sick when you take antibiotics, so we can't just give people a higher dose of antibiotics for an infection. So this is a serious problem. And exacerbating that problem, again, is that, when we take antibiotics, when we give antibiotics to animals and in the food industry, then these end up in our water systems. So it's been estimated that about 30 to 90% of antibiotics after going through our system still have this therapeutic potency. So they're getting into, they go through our sewer systems, there's water treatment that happens. And so what we've been working on as one part of our research is looking at antibiotics in wastewater, and then we can inform the wastewater treatment facilities that, okay, you need to do another treatment process to remove these antibiotics before you discharge into the ocean. Another issue that we're looking at is environmental sensors. So as I mentioned earlier in my title slide, that we have all these processes, agricultural processes, industrial processes, electronic waste, that end up leaching heavy metals into our water system. So we also want to be able to monitor for the presence of these toxins. And so because our sensors can detect well below the EPA regulatory limit, but because we can detect and quantify well below those levels, we can actually provide early warning that these toxins are getting into our water system. So with that, again, these expensive lab techniques, namely mass spectroscopy, prohibit us from monitoring water in real time. So again, we're trying to develop field deployable sensors that are low cost. The reader maybe costs a few $1,000s, but the sensor chips themselves are only a few dollars. So you can more frequently monitor the quality of water. So these are actually UCI students that were monitoring water during the COVID pandemic to look at, that was one way to determine the infection rate that was more reliable, is looking at how much of the virus was actually in the sewage system. Okay, so to go through my talk, I'll just give you a brief outline. So I'm going to talk about some of the engineering and technology challenges to develop this sensing technology. And then I'll go through the applications. So one that I mentioned earlier is the early detection of bacterial infection. So looking at these chemical signals to show us that a biofilm is forming and there's an infection. And then also looking at prescribing the right antibiotics for the treatment of that infection. So these are called rapid antimicrobial susceptibility tests. And then I'll end with talking about the application of water quality sensors. So to give you an idea, so I mentioned we use antennas to look at light, electromagnetic waves that are scattered from molecules. So this is called Raman scattering. So basically we have a molecule, we can shine light on the molecule. Some of the light will just scatter directly, but some of that light will lose a little bit of energy when it interacts with the molecule, and we call that a Raman scattered signal. And so what we do is we look at how much energy is lost as it interacts with the molecules, and we get these peaks. And so what these peaks tell us is they tell us the vibration modes that are present. So we can think of this as like a fingerprint, right? So we get this complex pattern. So we can do simulation, so we can understand what each of these peak corresponds to. And then here are just some movies that we made from the simulation showing that the blue curve at 989 wave numbers is what we call a breathing mode. So the ring just kind of goes in and out. We have stretching modes for particular bonds, the amide bonds there. And we have some ring and plane bending modes, right? So all these summed together will tell us if we have this pentoic anhydride in our system. So, but the problem is that our signal to noise is really low. So typically we need, very high concentrations of molecules to be able to determine if they're present using this technique. So some time ago, this group in the 70s determined that if you roughen a gold surface, so now you have these nanostructures on the surface, you can actually see a higher signal. So they actually just did an electrochemical process. They roughened a silver electrode, and they saw a higher signal from this molecule pyridine. And then about 3 years later, the group of Van Dyne came up with a theory for why this happened. And so I think it's kind of interesting sometimes to look back on the history of how technology develops. Like, for example, technology that was very important for us recently was the COVID vaccine. So the first mRNA vaccine idea was in the 60s. It took about 10 years before they actually showed that you can actually get this messenger RNA into a cell. So that's the 70s. But then they weren't able to make a vaccine for a mouse model until about the 90s. And then 2013 was the first vaccine that was used for rabies in humans. So you can see how long it takes for a technology to develop. And then, even with the proof of principle, it wasn't, it was kind of expensive to manufacture. So it wasn't until there was a global market that needed this mRNA vaccine that was actually manufacturable. So we're seeing now with the issue of antibiotic resistance becoming one of the top 10 world health threats. Typically, you need to make a lot of either vaccines or devices, right, to make it, the investment in manufacturing, worthwhile. So we kind of have the same idea here with surface-enhanced Raman scatterings. It's been a technology that's kind of been out there, but it's had some issues. So as I mentioned, over time, people understood a little more in detail what exactly was happening. So if you have two metal objects in the vicinity of one another, so this was this paper in the 80s, you have a nanoparticle next to a metal surface, and in between you get something called a hotspot. So as I mentioned, we look at the scattered light from our gold antennas here, and if it's right at resonance, so typically, like if you see the movies with bridges, failing in a windstorm, right? So if you get the right resonance of the excitation of a bridge, it can start having this oscillation. So similarly, the electrons in a metal can start oscillating in response to being excited with an electromagnetic wave. So we call that the local surface plasmon resonance. And what happens is, so once you have these oscillations, they oscillate out of phase, the two neighboring nanoparticles. So effectively, you get this what we call a hotspot, this region of a high electromagnetic field, which electric field squared is the intensity of light. So you can think of it as having a lot of photons localized in this hotspot. So there's a little cartoon. So in this region, rather than just having a few photons, so we have a lot of photons there. So we have more photons available to scatter with the molecules. So we can start detecting molecules at lower concentrations, because I mentioned in the prior slide that the scattering event is very rare, so we need a lot of molecules to have this scattering event. But surface-enhanced Raman scattering turns it around, and so rather than having a lot of molecules, we just create a lot of photons. So then we can see molecules at much lower concentrations. So here I just show a recent paper, relatively recent from these other papers, just showing now we understand very well, the origin of these hotspots. We can even understand quantum effects, when we have tunneling between the particles, and you can see once we get quantum tunneling at these separations below around 3 angstroms, and we kind of ruin these dipoles that we have here, and so we lose our enhancement. So you can't just keep on getting them closer together to get this enhancement. So I was watching late night this past year, and hopefully it will come back. Hopefully the writer's strike will end at some point soon. But I was very excited when I saw Stephen Colbert talking about surface-enhanced Raman scattering sensors. He couldn't pronounce it, but I was still happy that it was on TV. So we can look at things like, lung cancer, infections in the lungs, like looking at the molecules that come that are in someone's breath. It's been demonstrated with Raman scattering. It was used also to detect the presence of the COVID virus, influenza virus. And so here's a diagram showing, so there's many applications for Raman scattering here, right? But unfortunately, there's still some engineering that needs to be done and technology development. So this is a paper I published about 10 years ago now, just showing that these signals from a SERS, surface-enhanced Raman scattering, is very sensitive to the gap distance between these gold nanoparticles. So you can see if we have a 1 nanometer gap versus a 2 nanometer gap, your electric field intensity changes by a factor of 3.5. And so since we're looking at the scattered light, so remember light is E squared, so your signal can vary dramatically, right? So a 3.5 change in the electric field, is a 3.5 squared, but it's even worse than that because we're enhancing the photons interacting with the molecules and we're enhancing the photons being scattered from the molecules. So actually our intensity is e to the 4th power approximately. So 100 to the 4th power versus 350 to the 4th power is a very different number. So a big problem is that, our signal is basically more sensitive on the distance between the molecules, or between the nanoparticles, than the concentration of molecules. So this has been a big hindrance from using this technology to quantify the number of molecules present. And so one of the big issues in terms of detecting infection, right, there's bacteria in our mouth, there's bacteria already in our lungs, right? We're already colonized with bacteria, so we do need to quantify the amount of bacteria present even to detect an infection, not only just detect its presence. So that's been a big issue. So one thing that we did recently, so we've been working on coming up with new ways using chemistry to assemble these gold nanoparticle antennas. And so we've actually come up with a simple system. We can make a little capacitor and a microfluidic channel. So the silicon and the indium tin oxide are 2 electrodes. And we can do this in solution. And so if we apply an AC signal between these 2 electrodes, we can get fluid flow and motion of gold nanoparticles in this microfluidic channel. And if we look a little closer at this channel, is we can force chemical reactions to happen between the gold nanoparticles, and we can form a molecular linker. So here's an example of chemistry that we've developed where we use carboxylic acid functional groups. They will link and form an anhydride bond between the nanoparticles. And so now we have a very precise gap spacing. So we have something, it's about a nanometer gap spacing. Here's an electron microscopy image. Our next step was then to say, okay, well, how well do these sensors work? So we just used basically a dye molecule that has a very high Raman signal and said, okay, can we detect this dye if we at the single molecule level? And so we did an experiment. So first, if there's no dye present, we're going to have background noise because these gold nanoparticle antennas are very sensitive and they're scatter light. So since we have a polymer underneath and silicon underneath, we're going to see those signals. So that's our background noise. And the vertical bars are where we would expect to see the rhodamine 800 signal. So then if we put about a nanomolar, 10 to the 9 molecules per liter of liquid into our system, we're at about at the concentration where we would expect a non-uniform coverage of dye on our surface. So here's just a little image showing that, okay, some areas are just going to be bare gold, and then other areas are going to have dye molecule present. So in the 2 spectra, on the top and the bottom of this, we can see on the vertical lines, there's like little peaks where we would expect to see the rhodamine signal. But in the orange one, there's nothing uniform in that signal. So we can say, okay, is that region, is there no molecule there, or we just don't have sufficient signal enhancement to see the molecule, right? Because normally we would need something on the order of 100,000 molecules to be able to see a signal. So having one, it would be insufficient without these signal enhancements. So we used a reporter molecule to show us that, okay, if we look at the SIRS intensity across the surface, it's pretty uniform. So we don't make these sensors in a clean room, we just make them in the fume hood. So these little white spots are probably dust particles. But generally, our signal enhancement is something on the order of 1 billion across the whole surface. And that's basically the signal enhancement that you need to get to the single molecule detection limit. So this was a record in terms of performance, a large area nanoscale sensor that gives you a reproducible signal on a device relevant length scale. And so we published this, and then it had a write-up in Nature Nanotechnology talking about the significance of the work. So we said, okay, so now based on the fact that we see that we have signal enhancements across the entire surface, then we can assume that if there's no signal, that means there's no molecule on the surface. So then we use machine learning to analyze the signal across a sensor surface, and then we can say, okay, can I quantify how many molecules are present at these ultra-low concentrations? So we were able to detect down to 1 femtomolar. So a femtomolar is that we have 1 rhodamine molecule for every 10 to the 15 water molecules, right? So very, very low concentrations of rhodamine in our system. And we compared to a commercially available sensor. So the detection limit of the commercial sensor, you can see, is about 100 attomolar, so they can detect whether rhodamine is present or not. But you can see that the signal is because of the lack of reproducibility, the lack of the control of the gap spacing between the gold nanoantennas. Basically, you could just detect if they're there, but you can't quantify. Where our sensors, because of this reproducible signal, are able to quantify all the way down to 10 femtomolars. So that's, like I mentioned before, so when we want to use them in applications, this ability to quantify is really important. So these signals that we see, right, we just see a bunch of peaks here. So thinking, similarly, computers are used to differentiate different people's fingerprints. So we can use a similar type of algorithm to differentiate the spectral fingerprint of different molecules. So just a quick background on machine learning. We can use classification, right, which tells us if it's one thing or another thing, right? So do we have a picture of a cat or do we have a picture of a dog? So the way algorithms are trained, they're shown a lot of different pictures of cats, a lot of different pictures of dogs, and you tell the computer these are all cats, these are all dogs, and then the algorithm learns, like, okay, what's the difference between a cat and a dog? You can do semi-supervised learning where you have some training and then you throw some unknown pictures. You don't tell the computer whether it's a cat or a dog, and then you see where it classifies it, right? So if you wanted the algorithm to sort out all the cats versus dogs in your database, and you would use this semi-supervised learning method. And the other method for quantification is called regression. So you can have things that are in between, right? So rather than just having cat dog, you can have something that's partially dog, partially Elon Musk, and it'll tell you how much of one or the other is there. And so these are the kinds of machine learning algorithms we need for quantification. And so here's just where we quantify the amount of bacterial metabolite. So another advantage of this technique of being able to control the chemistry between our nanoantennas is now we can actually put in molecules that we want to detect, and they can react with our nanogap. So then we can kind of hold them in this hotspot region where we have a lot of photons, so it makes detection more easy. So here's just, again, an example. So we can look at the spectra of the reactant and the product, and we can see the change over time. So here's our experimental data, matches the simulation for this product forming over time. And the reason why this is important technologically is, if you notice here, so our prior work for rhodamine 800, there was no affinity with the nanogap chemistry, the chemistry between the gold nanoparticle antennas for the rhodamine 800, but we have affinity for this 2-aminocetophenone. And so we can see that our precision, right? So precision is how well you can differentiate two concentrations from one another. We can see that the sensor precision is much improved, like we can't really see any error lines between our particular concentration. So it helps us a lot, and it also helps us with detection in complex media where we want to detect certain molecules, but be able to ignore the background. So just a quick recap is that, so what we've done to overcome these challenges for using surface-enhanced Raman scattering, in actual applications is we've been able to define nanogap spacing between our gold nanoparticles and antennas, and this gives us a reproducible signal, and it also allows us to control interactions with the molecules we want to detect in solution. And so this reproducible signal allows us for analysis with machine learning. Our data sets are on the order of several hundred to 10,000, right? And the analysis technique usually takes us about a half a second to acquire a data point, so we can, get a reasonable data set in a few hours. Because we are using machine learning, we can think of it as mimicking our olfactory system, right? So in our olfactory system, we have receptors that are sensitive to certain chemicals, and so we can use this combinatorial information on which receptors get activated to determine which molecules are present. So I kind of give the example, if you go into an Italian restaurant, you can smell the cappuccino, you can smell the pasta sauce, you can differentiate all these odors. You have this complex background of odors, but you can differentiate these molecules, right? These molecules are activating your odor receptors. And so similarly, we have our gold nanoparticle antennas. They have a particular chemistry, so they can interact with different molecules. And then we can take this information, this combinatorial spectral information, and decode it with the machine learning analysis and determine what molecules are present. So we can go forward with that. Oh, and then this was kind of a fun fact. I found this paper. So the grasshoppers have a very sensitive olfactory system. So they can smell, like if they have a mate within I think it's something like 100 miles, something insane. So this one group decided, okay, so can I figure out what receptors get activated on the grasshopper when it smells TNT, right, an explosive? So they determined that, so I guess they would put these grasshoppers to sleep, connect these receptors to their head, and then whenever this network was activated, then this little light would go off. So they they learned how the neural network of the grasshopper responded when TNT was present, so they made these grasshopper detectors of TNT. So a similar idea, just the opposite of what we're doing here. So I'll go with our first application. So this was our early application looking at biofilm formation. So what happens during the process of an infection, so you have bacteria that's present, they start multiplying, and then they start sending out signals to one another. So if they form a colony, then they can make this polymer matrix surrounding the colony, so they're safe against other bacteria, things that they would compete for resources. And then they're harder to kill with antibiotics. So we looked at detecting pyocyanin, so what is called is quorum sensing, right? So as the bacteria, when they're isolated, they start sending out these chemical signals, and these signals are dependent on cell density, so that's why it's called quorum sensing. So once you have a high enough number of bacteria in the community, then they'll stop sending out the signal because now they have the polymer matrix and makes them a safe colony. And you'll see a plateau on the pyocyanin signal. And here's a pyocyanin molecule. And so what we found is that, so we had basically a region where we were growing the biofilm. We made a microfluidic channel, so we have liquid that goes over the biofilm and then goes into our sensor surface. And we were able to see after about 3 hours, we could detect pyocyanin signal. And then we could start quantifying how much pyocyanin was present at about 9 hours here. So this was our early work. And then I just list the different escape pathogens. So these are the typical bacteria that develop this multi-drug resistant profile. So they're important to be able to detect. So the idea here was, for example, people who are intubated in the hospital for long periods, they may end up with a lung infection because a biofilm may grow in their lungs due to the presence of this tube. So you can imagine just rinsing the intubation tube as it's changed and then determine if they're coming down with some type of infection early on. So we said, okay, so if we can detect the presence after 3 hours, does this help us? And so we compared, so if we treat the Pseudomonas colony with a typical dose of antibiotics to kill the colony, we can see that after 6 hours, if we treat it, we can kill a large fraction of the bacteria. The same with 10 hours, but if we end up treating the bacteria after 15 hours, then we kill less bacteria with the same dose of antibiotics. So they're starting to get this resistance to antibiotics. So this gives us the ability to detect an infection before the bacteria have this greater resistance to the antibiotics. But speaking with physicians, that's not enough. You also need to know the right antibiotic. So as I mentioned earlier, getting the right antibiotic to the person is important, right? So there are times when you go to the doctor and they say, go to the pharmacy, take this antibiotic, and then they call you 3 days later and say, no, that was not the right antibiotic. Come back and get this other antibiotic instead because the one you're taking isn't going to work. So the way these tests typically work is using PCR. So it takes about 1.5 to 2 days to get these PCR results. And so they require knowledge of knowing what genes lead to resistance. So here's some typical types of infection that are pretty widespread, and these are the antibiotics that you would normally be prescribed for these infections. But these are genes that are known to lead to higher resistance. So there could be a mutation, there could be gene transfer through some kind of plasmid, direct gene transfer between bacteria. So they can have these different ways of modifying their genes that they become resistant to the antibiotic. So it's not necessarily so reliable. So typically, if you go to UCI Medical Center, they're going to use these growth assays, right? They're going to take bacteria, they're going to culture it, and then they're going to expose it to antibiotics and see if it kills the bacteria. So this takes even longer than 1.5 to 2 days. This is usually 3 or 4 days that would take to learn whether this antibiotic will kill the bacteria. So this is what these growth assays typically look like, right? So you treat it with some dose of antibiotic. This is looking at trying to figure out what dose you need, this minimum inhibitory concentration, so how much antibiotic you would need to kill the bacteria. And see, once you get to where you kill about half the bacteria, that's what we call the MIC 50, where half of the bacteria will die due to the dose of the antibiotic. But this also is very long. So, but one thing so going back to the PCR, so we can look at genes, right? So we kind of use these symbols in the prior slide to show different mutations of genes in a bacteria, right? So the wild type is the most common bacteria that you're going to find of that particular species. So E. coli has many strains, and the wild type strain would be the most common. And these are mutations that can happen in the genes. But you can see that the mutation, the blue mutation, you have the same response to the antibiotic. And the red mutation, you can see, okay, it's something on the order of 10 times higher need for antibiotic to kill the bacteria. But if you get these two mutations together, now it becomes 30 times higher dose of antibiotic to kill it. So this is kind of unpredictable. And as I showed earlier in the slide with this very large Petri dish, like new resistance mechanisms are evolving all the time, right? So we want to keep this so we call phenotypic analysis, right? So phenotypic is the expression of a behavior, right? So just, people can look at their genome and say, do I have, the probability to develop cancer? And you can say, yes, based on your genome, there's like a 90% chance that you'll get some type of cancer. So those are the genes. But if you don't have cancer, right, you don't have the phenotypic expression of those genes. So that's the difference. So we say, okay, well, the growth assays give us the phenotypic, is this actually being expressed? And the genes just tell us the potential for expression. So the phenotypic response is the most reliable, so it's still kind of what we call the gold standard in antibiotic resistance. So fortunately, a lot of work has been done, and so we work a lot with E. coli. And so just an interesting piece of information, a lot of the biopharmaceutical industry relies on E. coli as a mini cell manufacturer, right? So basically a lot of pharmaceutical drugs are, you modify the genes of E. coli to produce a particular drug. So it's very well understood. So if we look at here's a review article that was interesting, just showing how much we've learned over time. So basically, in the early 2000s, we started understanding a lot about its metabolism, the expression of certain proteins. And then recently, as I mentioned at the beginning of my talk, it's a stress response. So the stress response is what tells us whether the E. coli is susceptible to an antibiotic or resistant. So we use that in our research, all this understanding, to say, okay, now I can use that stress response to tell me whether my antibiotic is going to kill the bacteria. And so I mentioned, so typically we have to wait, maybe anywhere between 2 and 4 days to figure out if our antibiotic is going to be effective. So what that usually means is that doctors don't wait. They just prescribe it and you get, the antibiotic, and we get more antibiotics in our ecosystem, which is not good. So we can say, okay, so how can we kind of short circuit the time to determine if a bacteria is going to be resistant or susceptible to antibiotics? So this is some interesting work that was done some time ago, and we use the basis for our work is that, so we have 3 different kinds of antibiotics, and they have different ways of killing bacteria. One way is maybe stopping the bacteria from reproducing, so your immune system can take care of it. Others just directly cause the cell wall to disrupt, and then the bacterial cells die. So we have different killing mechanisms, but what this group found is that early on, there's a change in metabolism that is very similar for the different antibiotics that we see here. And so we said, okay, well, if that's the case, can we detect those metabolic changes very early on with our sensors? So we found for E. coli and Pseudomonas aeruginosa that we can start detecting these changes as soon as 5 minutes. So this is a machine learning algorithm that's a classification algorithm. It's called a confusion matrix because it's a little confusing. But basically, it tells us that I can classify the chemical response of the bacteria with 99.5% accuracy, so about 99% accuracy, like if at the initial point before I give it the antibiotic, and then 5 minutes later, I can start seeing a change in the metabolism. So then now we have this idea, rather than 2 days, we can start having antibiotic susceptibility tests, and maybe it's an hour to prepare the sample, and maybe at max another hour to analyze the data. So then within a couple hours, then we can determine the right antibiotic. So either during your visit to the doctor's office or the time for you to get to the pharmacy, then you can know the correct antibiotic for your infection. And so just a little bit of information on how we do this, right? So we look at these spectral signatures. So these bacteria, like now they're just emitting all kinds of chemical signals. So the data is very complicated. And so we can classify the spectral response, how similar they are. So this is called a variational autoencoder, the type of neural network that we use to differentiate the response. And the nice thing about the VAE is that also can decode, so then it can tell us like what's different, right? So if we say that red is resistant to antibiotics and the brown is susceptible, then we can look at those spectral features and we can say, okay, well, what's the difference between those features? So it gives us, it's not just a machine learning black box. So with the autoencoder, we can look at no antibiotics as our control. Carbonicillin is where it's susceptible, and rifampicin, this particular, this is Pseudomonas aeruginosa, is going to be resistant. So we can see that we can kind of classify a different response. We can start distinguishing between if it's going to be resistant or susceptible, but we can see that, our classification isn't the greatest. There's still some data points that overlap. You can see the blue and the red, for example, there's overlap, so we can't really differentiate them. So we said, okay, well, since we get some information about what is actually changing in the bacteria, we say, okay, well, let's look at some potential metabolites that are leading to differences in response. So in order to get this training data, we have to culture the bacteria and look at the response to the antibiotic after half an hour, after 2 hours. And we say, okay, what's different? But we said, okay, well, since we can kind of see that we have a lot of changes in this gray highlighted region on your upper left, we can say, okay, well, let's look at metabolites that have features in that region, and let's just buy some metabolites for sigma Aldrich, and let's use them as additional training data. So we don't have to culture that. We can just do those experiments right away. And so when we add that to our training data set, so this is what we call data-informed transfer learning, then we found that we can really differentiate between the susceptible and the resistant response. You could say we have a few miscategorized responses, but now we have 99.5% accuracy, where early on we had something in like maybe 85% accuracy. So talking to physicians, we've done a lot of customer discovery interviews, and we found that, we asked physicians, okay, if you had a technology that could tell you the right antibiotic, what kind of accuracy would you need? And they were like, it would have to be like nearly 100%, at least 99%. And so this was probably around 8 years ago, and we're like, that sounds very hard. But through all this development of understanding how to make very sensitive and reproducible sensors and how to bring in machine learning, we're able to show that a few years ago that we can achieve that accuracy. And what's also interesting, as I mentioned, we get some information about the features, so we can label what kind of peaks. So these are some of the metabolites and nucleotides that are changing in response to the antibiotic treatment. So it tells us what kind of biochemical network response is happening in the bacterial cells. So it gives us information. So you can also do some fundamental studies on bacterial response to antibiotics, which could be important for developing new antibiotics to deal with some of these resistant strains. So I'll go ahead and move on to our water quality sensors now. So not because we still have a lot of antibiotics in our water system. Again, we took this commercial SIRS sensor that's available. You can buy yourself. They're called SIRS Straits. And they use these little gold nano fingers. And so when you expose these nano fingers to a liquid, and then you evaporate the liquid, they start collapsing and they trap molecules in these gaps. So these are probably the most sensitive SIRS sensor that you can buy on the market now. And so we compared it to our sensor that just works directly in water. There's no need for evaporation. And so we looked at this particular antibiotic, quinolin, to see like, okay, how does our sensor, stack up against what's commercially available? And so here's some data. So when we just look at the antibiotic in deionized water, so it's just pure water with antibiotic present, we see very similar performance, so maybe a slightly stronger CO signal with the commercial SIRS substrate, but both can detect it down to 5 parts per billion, and both can quantify at these relevant concentrations. But then when we go to wastewater samples, because we have our nanogaps kind of occlude any larger molecules, and wastewater is going to have all kinds of particulates and things in it, we can still detect the quinolin, where the nano finger samples, you can see that the intensity is all over the map. And so you don't have reliable detection. So again, it kind of is the example of having this controlled chemistry that we're only sensitive to the particular small molecule that we want to detect. So, and then here, just kind of doing again, using a machine learning algorithm to determine the concentration. This is just a simple regression algorithm. We can see that the limit of detection of the commercial substrate on your right is very high, right? Because the differentiation from the background signal is not good at about a half a part per million. But here our sensors go down to parts per trillion levels here. So we can start detecting at very low concentration. So again, this idea of reproducible response is really important. Okay, so then I'll get to the final application. So I mentioned this understanding of the stress response of bacteria. getting back to the main title of the talk is then saying like, okay, so since we understand that bacteria, whenever they're kind of threatened by some kind of toxin and antibiotic, but also things like heavy metals, they're going to send out some chemical signals. And so we said, okay, so can we use these bacteria then to look at wastewater samples? So heavy metal ions are particularly hard to detect, right, because it's just basically an atom, right? So you really need very sophisticated technology to detect these ions at low concentrations. So it can be very expensive. Now there's a lot of commercial available ways of detecting at higher concentrations, but there's no quantification. So we said, okay, so we take the bacteria, we culture the bacteria, and then we could put some water samples in with the bacteria that have these toxins present. And then we, what we call lyse the cells, break open the cell walls, and we can look at the metabolites that are produced in the cells. And then we can take these chemical signals and figure out if water is safe or if it has toxins present. So past work using similar approaches usually required one to functionalize your surface, your gold nanoparticles with an enzyme or an aptamer or some other chemistry. But that requires you knowing that, okay, I want to detect arsenic, which isn't necessarily a bad thing. Maybe you're in an area that's known to have arsenic in the water and you want to detect that. But you would still require a knowledge of what you want to detect. Whereas we're saying that with our process, and we don't need to know if arsenic or chromium or other types of contaminants are present. We can actually use this network response to actually differentiate the contaminants without surface functionalization. So we can see again, so a classification model. So we can say, okay, if I put different amounts of here, in this case, chromium in my water, and then look at the bacterial response, can I differentiate it? So the control is no chromium in the water. So we can see at about 0.68 picomolars, we have, some overlap between the control and the metal concentration. So we say, okay, well, I'm not going to count that as a being able to detect. But once we get to about 6.8 picomolars, then we can differentiate very well with the control. So above 95% accuracy, we can tell if there's metal in the water or not. And so we define that as our limit of detection. And similarly for arsenic, we can do the same, right? So if we define our limit to where we have 95% accuracy, then we can say, okay, our limit of detection for arsenic is a little higher. It's about 0.5 picomolar. And so this kind of basically scales with the toxicity. So if you look at EPA regulations, the maximum contaminant level of arsenic is about 10 times lower than chromium because arsenic's more toxic. So we can look at other traditional biological methods to look at it. So optical density, I mentioned this earlier in my talk, where we can look at basically when cells are in solution, they're going to be swimming around if they're alive, but as you kill the bacterial cells, they're going to fall out of solution, and so then the solution will be less opaque, and then you can use that as a measurement of how many cells you're killing with a particular process. So looking at this measurement after 2 hours, let me take a drink of water. So we can see that we can't really differentiate too much of a change in the number of live versus dead cells, right? So this would be the level where you're actually killing cells, which you wouldn't want that much chromium in your water. And so it requires about 340 micromolars of chromium to be present for one to be able to detect it. And similarly for arsenic, we don't really see a change until about 100 micromolars. So much, much higher than what our machine learning analysis and chemical signals from the cells are far, many orders of magnitude lower. And so we also compared a regression model, so that was a classification model, so we can say, okay, can we actually quantify? So as I mentioned before, so for this particular example, our data set was about 10,000 spectra at each concentration. So we have very big data sets, so we use this box plot here. So basically, within the box, that's like 50% of the data has the same response. So we have very nice reproducibility. There's a few outliers, so we're going to have some biological variability. The bacteria don't all behave the same. So we can see that we can basically quantify the amount of chromium and arsenic present here. So we follow this nice line of what we expect, how much arsenic or chromium we put in the water, and then how much our model predicts is in the water. So we have this nice one-to-one correlation. And so we can get a limit of detection, which highlighted, which matches our support vector machine classification model, and we can get a limit of quantification where we can tell exactly how much is in the water at about an order of magnitude higher. And so the other thing I mentioned is this not needing to know in advance what kind of toxin is present. So again, this is looking at the features, right? So these are the spectral features that are changing. So we can see the either bright red or bright blue are the features that are changing in our spectral data most significantly. And if we look at that, then we can differentiate whether chromium or arsenic is present in the water. And so we can look at these features and we can see what metabolites correspond to these particular scattered frequency features, right? So these correspond to some vibrational mode that tells us what kind of molecule is present. So we can learn something about, again, the network response. So again, all water sources are going to have different background contaminant. So since we do this in the lab, we said, okay, well, how good would this be in the real world? Because with different background contaminants, so then our algorithm won't really understand, the chemical signals that we see. So can we do something called transfer learning? So we can train our models in the lab with clean water, and then we can say, okay, well, let's just take a water sample in the field and see if we can also predict if the water is safe or not. So we took our trained model in the lab, and then we were able just to take a subset of the data at particular categories, and then train unseen tap water samples at different concentrations, right? Because you may not know the exact concentration of the contaminant in your water sample. And we said, okay, so with that, with 99% accuracy, we're able to differentiate if the water was above or below these regulatory safety limits. And so we found in order to do this, we only had to take 80 spectra per class because we've already trained the model, and now we just have to kind of train it, kind of tweak it a little bit to understand this different water sample. And so for tap water, we did 80 spectra per class, so that's about 40 seconds of acquisition time. So this kind of enables going out to the field. You can basically go out to the field, you take your unknown water sample, you might spike in some impurities to fine-tune the model, and then you can take your unknown and then determine if it's safe in terms of the amount of toxins present. And similarly, we said, okay, well, tap water is not so problematic, but wastewater, as you saw, has all kinds of background contaminants from the antibiotic work that we did earlier. And so similarly, we said, okay, well, let's just use the same approach. We're going to take a subset of concentrations and fine-tune our training of our model with these concentrations. And here, with greater than 96% accuracy, we were able to determine if the water had toxins above or below the regulatory limits. And again, similar, so very short time periods, we're able to fine-tune. So we can see, taking these samples, the spectrometers, the readers for looking at the spectral data, or, maybe something in the order of this large, so you can take them out to the field, and you can do this kind of machine learning and analysis once it's trained on basically, a laptop computer. So these are definitely field deployable. So just, I'll put up my conclusion slide, basically coming up with a process. It's a solution phase process for assembling these sensor surfaces. We can use processes that are used in terms of manufacturing plastics to make these sensor surfaces so very scalable to, making, hundreds of thousands of sensors, manufacturing these with very reproducible response. So basically, you can buy, like if you had a lab clinic and you bought a reader for maybe $5,000, the sensor surfaces themselves are a few dollars, will perform as well as a $500,000 mass spectrometer that you would find in the hospital. So it makes it much more feasible for these surfaces to be used, globally for many applications. And with those, we've shown the ability to detect infections before the bacteria become resistant to antibiotics, the ability to determine the appropriate antibiotic to use for the infection, and then for use as water quality sensors. So again, using machine learning and using these kind of transfer learning approaches where we can fully train a model and then just use a few samples to kind of fine tune to be able to use this machine learned model on unknown samples. And so, well, thank you very much for your attention. And so, open for questions now.